So understanding genetic maps is nice, but really what we wanted was to know the whole sequence of the whole genome. So we're going to talk about a couple different ways you can go about sequencing of genomes, and what we really wanted to achieve was a whole genome sequence. So figure out all of the DNA sequence that is in an organism. Now previously we talked about Sanger sequencing. We use specific primers and especially modified PCR reaction with fluorescently labeled ddNTPs to read out the sequence of DNA. This process works pretty well. I'm not going to say it works 100% at the time. On good days, it works 9 out of 10 times. So that sounds good, but when you're doing a lot of sequencing, that's an appreciable failure rate. The big problem is that you can get, at best, about 1,000 bases of DNA sequence out of this Sanger sequencing read. Sounds good, right? 1,000 bases. But let's consider the human genome is 3 billion bases long. That means we need at least 3 million reads of this sequencing. Now, consider that one of these sequencing reads could cost $10. That is what, $30 million conservatively. You're not going to get it in that small of amount though. So what we really need to do is we need to do a lot of sequencing. And what we want is we want to create sequences of overlapping sequence that we can stitch together. So we're going to start thinking about how we could go about sequencing the human genome using this technology. So let's assume that there is some base underlying sequence, right? A chromosome. If we could sequence a piece of it, and another piece of it, and another piece of it, if these pieces overlapped, we could start stitching the sequence together there. That would be a good way of going about sequencing the genome. So we need some sort of physical map, and then we want to sequence pieces of that and form what are called contigs, or contiguous units of DNA. The Human Genome Project, the endeavor to sequence the human genome, started with this kind of concept. This was primarily funded by the U.S. National Institute of Health, the NIH. And in 1990, they basically set out a mandate that we want to sequence the human genome. The sequencing itself was done by about 20 universities around the world. They planned for it to take about 15 years, and they budgeted about $3 billion to do this, to pay all the people for all the equipment, the time, the materials, all of that stuff. And what they started doing was they took a chromosome and they chopped it up into what we call little clones, little pieces. And they started sequencing those pieces, and they tried to overlap those pieces and do little sequencing reads, kind of like chapters in a book and pages in a chapter, and stitch it all together. Get these clone contigs here. They quickly realized that this process was, although in theory useful, not really realistic. They weren't getting enough overlap, or it's just taking too long to do. So an alternative method was thought up. What if we take the genome, we chop it up randomly, and we just sequence those random bits? Then, using a computer, we try to stitch the pieces back together. Computers are really good at matching sequence and aligning things and doing these really repetitive tasks, right? So they were turning to computer programs to try to reassemble the DNA sequence. This is called shotgun sequencing, right? Because you're just kind of throwing it out there and seeing what sticks, right? Hitting different pieces. Shotgun sequencing breaks up the genome into random parts. We sequence the random parts, use a computer to put it back together, kind of like trying to solve a jigsaw puzzle. We call that assembling the sequence. Now, what's going to be the problem with this? Well, you see these overlaps, they're pretty small, right? You're talking 6, 7, 8 bases long. How frequently do you think those 8 bases occur in the 3 billion bases of the genome? They might happen many times. And there are indeed some places in the genome where we have repetitive sequences that are found throughout the genome, or a bunch of repeats in a row, right? So we had major problems with repetitive sequence here. Let's look at the challenge of this, right? Sequence assembly challenges. So we have here, it was the best of times, it was the worst of times, right? It was an age of wisdom, it was an age of foolishness. It was an epoch of belief, an epoch of incredulity. It was a season of light, a season of darkness, blah, 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 blah. All right, if we break that up into a bunch of random pieces, right? Cool, we can get a little bit. It was the best of, age of wisdom, it was, best of times, it was. It was the age of, it was the age of. It was the age of, it was the age of. We have repeats here. How do we know that this age of goes here and this age of goes here, right? There's problems with this. We can start putting it together. It was the best of, was the best of times, right? We can start doing this, but at some point we're going to start running into problems here. So, problem with the genome is it's actually more repetitive than Dickens, right? So we're looking at major computational problems here and ambiguities in there. So the human genome has its issues. We actually started smaller. We started with yeast, C. elegans, and Drosophila. All of these had their genomes sequenced to some extent. The yeast genome, really useful. Yeast is a single-celled eukaryote. C. elegans, a model worm species. And Drosophila, that formed our major basis for genetics. So Drosophila is really nice. It has 3 chromosomes. It was easier to sequence. Smaller genomes. But the human genome is big, 3 billion bases long. Like I said, Sanger sequencing, you're getting 500 to 800 bases, maybe 1,000 if you get lucky. And really what we need is coverage. We need to read each base in the genome at least 30 times to get high accuracy. The problem is that about half of the human genome is repetitive DNA. It might be remnants of viruses or what we call transposable elements. It's problematic. So we're trying to do these clones and these pieces and stuff like that. We have these huge rooms that have all these sequences in here. They're running 24/7, 7 days a week, at 1,000 bases a second, 24 hours a day, 7 days a week. That would take 6 years, right? That's highly optimistic. 6 countries, 20 different institutions. We're trying to brute force this, right? Huge computer clusters trying to do this. The Human Genome Project was not working. And this kind of came to a head because 2 people kind of faced off here. So this guy, Craig Venter, I'll talk more about him in a moment, he had worked at the Human Genome Project, and he realized there might be a better way to do this. So he started a private company. Francis Collins, on the other hand, was the kind of public face of the NIH-funded program. And Francis Collins, he's a physician, he's a scientist. He worked on the Human Genome Project, and he kind of became the leader. He was later appointed NIH director. I indeed actually went to a conference where I saw him speak as NIH director. Very influential individual. Craig Venter, on the other hand, he started at the NIH and then went into private industry. And he realized that there might be some money to make in sequencing the human genome. So he started a company called Celera Genomics that was competing against the publicly funded Human Genome Project. His goal was to sequence human genome bits and sell subscriptions to access this genomic data. It's got to make money, right? There are several, I would say, ethical issues with this, but it's his right to try to make money, right? So there was kind of a race to sequence the human genome at this point. This clone by clone, bit by bit effort from the human genome, kind of the tortoise in the race. And then Craig Venter at Celera, he did this whole genome shotgun sequencing approach and try and stitch it all back together. Now, he's kind of the hare in this. And there's kind of a unfair bit to this because Celera, being a private company, did not have to share their data with anybody. But the Human Genome Project, being publicly funded, did have to share their data with everyone. So the Celera company could benefit from this. Regardless of that, in 2003, they announced the completion of sequencing of the human genome, our draft sequence. I remember this very distinctly, actually. And the competition between Celera and that kind of faded away. There were several court rulings and things like that you couldn't patent DNA sequences and things like that, and it kind of dampened that. But they have a draft of the human genome sequence in 2003. All right, so what happened here? Well, it's 13 years. They're ahead of schedule. They thought 15. They were under budget. It cost about $1 billion, so $2 billion less than they thought. That's still a lot of money, right? But they had good high-quality sequence for about 90% of the genome. So when they said it was complete, it wasn't really complete, right? Like, there are gaps in there. That race between the Human Genome Project and inventor and his Celera company kind of went away, and they, didn't end up making much off this. Venter went on to do very, very interesting things. He's an interesting figure in biology. So kind of the concerns about sequencing the human genome, would it, violate everybody's privacy and blah, 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 that kind of faded away as well. But it really transformed the theory of this field of what we call genomics, the idea of sequencing a whole genome, and was very, very important for biology, medicine, and just societally. The Human Genome Project itself was actually composed of sequence from, I think, somewhere around 20 individuals, whereas Venter's Celera Genomics was actually sequencing his genome. So maybe a bit of an ego there, perhaps. So we got about 92% of the genome sequenced. That other 8% was not readable using the techniques of the time. It's too repetitive. We couldn't get through it. Parts like the centromeres or the telomeres were not super important, but were potentially important for certain medical conditions. So we still wanted that, but it would take another 20 years until we would finish sequencing the whole thing. So that last 8% took another 20 years to sequence. But new methods needed to be developed to do that. So we got a full, complete human genome in 2022. I remember this very, very distinctly. I was out of graduate school. Been a long time since that draft of the human genome. And we finally filled in all the gaps, right? But it's kind of like a puzzle where you solve the puzzle, but then it's like, okay, what do? We have all the sequence, but what does it mean? And that's what we're still going through. We're still trying to figure out exactly what it all means. Now, something that's really interesting that developed in that time was what we call next generation sequencing technologies, or NGS. I call these high throughput sequencing technologies because we can get a lot more sequencing. You're talking sequencing technologies that can give you 1,100,000,000 base pair reads, so 100 billion bases of sequence in a read. That means that now you could theoretically sequence a human genome for around $1,000 versus $1 billion. It's a pretty big shift, and that's really important when it comes to thinking about personalized medicine, understanding people's unique genetic code and how that might relate to like disease or things like that. If you want to learn more about these next generation sequencing technologies, take my Micro 250 class. We talk a lot about them. Okay, so what does shotgun sequencing have problems piecing together? Go and look at the options, think about this. So it would be repetitive sequences. Repetitive sequences are very difficult because we can't figure out how to stick them together right. Is it 5 repeats? Is it 10 repeats? It's hard to tell for the shotgun sequencing method. All right, this human genome, the whole genome sequence, we want to know the whole sequence of the DNA in an organism. Sanger sequencing gave us about 1,000 bases per read. We tried to get overlapping reads. That took a long time. Went to the shotgun sequencing method. We initially started with smaller organisms, C. elegans, yeast. I didn't mention Arabidopsis, the model plant system, which had a genome as well, sequenced. Wasn't until the next generation sequencing technologies that we could really figure out how to get through the whole repetitive bits. And we've got the price down to about $1,000 for a human genome. Okay, in the next section, we're going to talk about genomics and proteomics here.